Hello, good morning and good evening, based on your time zone. Today, I want to talk very briefly about ddPCR and how you can set up a very quick ddPCR for VSVG detection. In recent years, retroviral and lentiviral vectors become very important tools for delivering genes to different cell types in different experiments. And they are not only important or useful or used in labs, but also we use it for clinical trials to treat genetic disease, especially cell and gene therapy field. Even though we use safer second generation or third generation lentiviral packaging system, it is still important to ensure there is no leftover of genetic materials from lentiviral or vectors that could lead actually another secondary cancer or other complications which is unwanted. And to check for safety from several approaches, we can test for VSVG, which is a very key component in making this viral vector. Uh, so, since we need very high accuracy, I always recommend using ddPCR to detect these VSVG. And I'll show you very quickly how you can set up a ddPCR since we have a lot of myth and a lot of complications about ddPCR. Many people I heard they're afraid about ddPCR and it is not very common to be honest in academia setup. So I will just show you very quickly how you can convert or how you can just do a ddPCR using your general qPCR or general PCR knowledge. So ddPCR, which is digital droplet PCR, it is a very highly sensitive technique for detecting and quantifying nucleic acid. It works by partitioning sample into thousands of droplets, each droplets then undergoing going for individual PCR amplification, and these allow for precise measurement of DNA or RNA without using any kind of standard curve. So, which is a very good advantages of ddPCR because of its resolution is really high, and you really do not need a standard curve, and it is ideal for detecting very low level targets or small differences in gene changes or genes expressions or mutations. So this is a very typical ddPCR workflow. You add your mixes, you add your primers, probes, samples, and then in ddPCR we make droplet. So previously we used to use a manual method to make droplets, but now probably every lab or most of the lab use automated droplet generator. So which is exactly they make 20,000s of droplet for each of your experiment, like each of your wells. And after droplet generations, we go for regular PCR, and we follow the regular PCR techniques like the annealing temperature. We change for a good amount of the PCR applications to avoid non-specific binding. But I'm not talking about the details how you can set up a good PCR. You will follow each of these methods, thumb rules, techniques to continue your PCR. And when your PCR is done, you need a specific instrument to read each of the droplets, which is a QX200 droplet generator reader we can use. And ddPCR is a binary results. It will give you, it can detect one or zero, whether you have your target one or you don't have it. So I'll talk details, not very details, but I'll just come back these slides and show you each of the steps how you can do very easily. So Since we're talking about the VSVG detections, you start with the VSVG sequence that you want to detect. And of course, you have to design a primer sets like the forward primer and the reverse primer. And again, I'm not gonna discuss the details of primer design. You know how to do that. It's just a regular PCR. You will follow the thumb rules I added to websites, which is very, very good. They explain very nicely if you're a beginner. So you can follow this thumb rule and design a primer. And in ddPCR, we always use hydrolysis probe. So we do actually the probe-based PCR. And for a VSVG, I will use the default color FAM. So if you use the Bioware system, the FAM is the first color you must need to choose. And if you want to do multiplexing, that means if you want to add more color, then you can use hex or VIC. But FAM would be the first color you choose. So I choose FAM color conjugated with my hydrolysis probe to detect VSVG. And like every other experiment, I will also choose my reference gene. So just to give you example, like educational purpose, I'm showing you that I am taking GAPDH. And since this is my reference gene, definitely I have to also design the primer sets and the probe sets. And I just mentioned that we do the hydrolysis probe based ddPCR, and the family is the default color, which I have chosen. So now I'll choose another color is hex. I could choose VIC. There is no benefits choosing hex over VIC. And the next thing, you make sure that your one sets of primer doesn't have a cross reactivity with your another sets of primer or your another target of gene. I mean, the VSVG primer shouldn't have any cross reactivity with GAPDase or vice versa. I always recommend to use restriction enzyme because I am probably a little bit finicky, and when I use the restriction enzyme, I always have a better results. You can use this link. I'll just show you very quickly, and following this link, you can just make your restriction enzyme. So what you have to do, you will select your gene sequence, you'll copy it, and you will just paste it. This is just only one way I'm showing. You can use any other website. There is a lot of good website from different vendors. It's free, so you can use. So I just selected all of my sequence from VSVG, and I'll just submit it to see that which restriction enzyme is compatible with my sequence. That means which restriction enzyme can cut, or which restriction enzyme have a cutting site within my genes and which doesn't. So, and I'll see that these all restriction enzyme listed will cut my target sequence VSVG. And if I go bottom, I'll find also another list which actually doesn't have any interaction or doesn't cleave. You see the non-cutting enzyme. So I played with this and I found that hint 3 actually is the restriction enzyme which doesn't cut our VSVG. But I have to also check that hint 3, if I choose, it also compatible with my reference gene, which is GAPDase. So I'll just copy it and I'll just reset and I'll paste the sequence from my reference gene and I'll submit. So I know that I choose hint 3, which is is good for my VSVG. Now I'll check that hint 3 whether it's good for my reference gene or not. And you see that hint 3 is also good for my reference gene. So I will just quickly jump to my presentation just to show you that I can choose very fairly hint 3 restriction enzyme, which is compatible with my VSVG and GAPDH. You can play with your sequence and you can choose any restriction enzyme. There is not good or bad if it's compatible. And I always recommend to use 5 to 10 unit of restriction enzyme. And another big thing I always recommend to use the Proximar 188, but you must have to make sure that you do not use more than 0.01%. So you should use very low concentration of Loxamar it helps you because it helps you to reduce surfactant. It gives you the more stability in your droplet when you will generate your droplet. But if you use the more concentration, it actually can make problem in your real PCR reaction. It can work as inhibitor at that case. So definitely it's a very low quantity. Please make sure that you do right calculation. So the first thing we do, the ddPCR reaction setup, we make everything in mix in a one container. So I'll start with the ddPCR Super Mix for Pro. You can buy this from the BioRed. Since I'm showing the BioRed one, I'm not marketizing there. It's because just I'm using BioRed right now, so I'm just giving you example. But if you are Thermo Fisher guy, you can use Thermo Fisher um, or there isn't as well. And the second thing, you will add the primer and probe sets for VSVG and the primer and probe sets for your reference gene. And then the restriction enzyme, as I just mentioned, that is better if you use restriction enzyme, then you will have very less or no non-specific application. And I know people have a different opinion, but I use my reference gene with my master mix since I know that I will end up using my reference genes in all of my experimental well. So for better pipetting or to make our result uniform, I always use my reference gene with my master mix together. So next thing you have to use the ddPCR 96 well plates. There is a special plates by Red also sell. You have to buy it, and I'll show you a very simple way to set up the reactions. So initially you will use your master mix, which you just made with um, primer sets, probe sets, the restriction enzyme, the reference genes, and super mix, and you will use these master mix to all of your well. And then the first two well I will select for my positive control. And my positive control is the sequence of the VSVG, which we just talked in our previous slide. So you can actually send the sequence to the vendor and you can actually purchase the VSVG known sequence from vendor and you can use that sequence as your positive control. If you're in cell and gene therapy, you can use the same plasmid you use to transduce your T cells. There is a lot of way we can choose the positive one, but if you're unsure, you just can send your sequence to the vendors, and they will synthesize and purify the sequence for you, and you can use as a positive control. And the next two sets of experiment I usually choose for my sample, and you shouldn't use a very high concentrated samples. I'm not talking details about the sample quantity, how you can optimize it using nano drop, then using dilutions. I will not talk details on that, but you should use sample very low concentrations since ddPCR is very, very capable to detect very low concentration of um, your DNA. And then the last one is your negative control. So the negative control is you will use just buffer. Remember that in negative control, you have also your reference gene. So all of my experiments, I have a reference gene. So negative control have a reference genes and buffer. Samples have reference genes and sample. You mean sample means the testing sample, and the positive control means positive controls VSVG and also reference gene. And you'll transfer these plates into um, your automated droplet generator after sealing it. So you see that sealing one is your this is your the plate you just prepared, and you have to add a new plate where actually droplet will be collected. So remember, you are here this is step to generate droplet which will be generated by machine, but you have to put a new plate to collect the new droplet. And when it will make the droplet, it takes, if you're full plate, it takes approximately probably 40-50 minutes. And if we have very not full plate, it will make very shortly, like the plate I selected, it will be done in 10 minutes. Here is a one note. If any day for any reason, if you have the wells which you are not using. Say you are using all of these 2A to G and you are not using H. You must fill up the H with buffer or any water would be fine, but I would recommend buffer. The idea is that you cannot let you cannot make a blank. You must have always some liquid in your well if you select to well. So if I select column number 2, then I need to fill all the well in column number 2. I'm fine left blank 3, 4, 5 to 12 since I'm not using this column. But if I choose to select any column, I need to fill up all of the wells in the column. And the next thing we'll do the regular PCR. You can use C1000 thermocycler and you will do the PCR. And again, I said for PCR, you need to optimize your temperature based on your primer design. And the last thing, you will use droplet reader. So we use QX200 Doppler reader to read each of the droplet. And if everything goes fine, that would be your tentative results from your QX200 reader, QX Manager software you can use. And you see that in my x-axis, it's a hex channel or hex based result I should see. And in my y-axis, it's a FAM, so VSVG. So this is the plot I generated. This is the tentative result you would probably see after doing the VSVG ddPCR. So to see the blue dots left side um, top quadrant, that's your results from VSVG. And the green one is your reference results. So in VSVG is not good at that point because we don't want to see the viral particles or viral envelopes gene still in my product. So we shouldn't see uh, these results, to be honest, in the lab. We should see there is no VSB in my samples. That would be a then successful criteria to pass our CAR-T or gene therapy product. So typically we should see this is the results, and we should be happy if we see this kind of results. But if we see the VSVG in the results, That means our product is contaminated. We cannot use, or we cannot release, or we cannot like give this product to our patients, which is very, very dangerous. So I hope you learned something about ddPCR. I know this is not very details, uh, but you can use as a very starting point. And if you have any question about ddPCR, you can always DM me. And this presentation is only meant for educational purposes, so it cannot be used as a reference to make anything in your lab. Thank you.